Austin, can you hear me? You're muted. Unmute you.
Yes.
Great. Okay. I understand you're going to be doing the introduction.
Yep, that's correct.
Alrighty. Well,
Do you want to handle the questions from the chat too, Austin? Is that okay?
Sure.
That'd be good.
Do you want those during the presentation or after?
After, I think.
Okay. Generally wait until the end.
Yeah.
All right. I'm going to let everybody in.
Okie dokie. Hi, Ann Pabst.
Hello, Andy Reid. How are you?
I'm good.
Oh my gosh, there's Lee Torres. Oh my gosh. Mr. McClellan is going to be coming in too as
Good.
our perhaps 100 Bio 358 students and certainly members of the lab.
I really hate you right now, Ann Pabst.
Hi, Andrew and I are here too.
Hello. This is fun. That's a very good image.
That's the waterfront of Beaufort.
Ah.
Hi, Andy and Anne and Heather. It's Zoe.
Hello.
Hey.
Zoe. I don't know.
These
You can
events
just-
are going to end up being one of the highlights, silver linings of this event.
Oh my God.
I know I'm
Andy,
trying.
you really kept this under wraps, Andy. I had to find out about it from my postdoc who sent it to me in a text message.
I really tried hard to keep this under wraps, and it went on something called Facebook.
Yep.
Hi, Ambika.
Hi, how are you?
I'm good. How are you?
I'm good. I'm good. I
Good.
actually just texted you saying I was here.
Okay, good. I can see you.
Oh, hang on.
All right, Andy, did you want us to get started?
I guess we should get started. It's that time.
All right. Well, welcome everyone and thank you for joining us. My name is Austin Allen and I have the pleasure today of introducing my advisor, Andy Reid. Andy's the director of the Duke Marine Lab and chair of the Marine Science and Conservation Division. He's also the Stephen Toth Distinguished Professor of Marine Biology. He did his PhD at the University of Guelph and a postdoc at Woods Hole Oceanographic Institute. Andy serves on the Committee for the International Recovery of the Vaquita and has served on scientific committees for the International Whaling Commission, the Marine Mammal Commission, numerous editorial boards, and is a past president of the Society for Marine Mammalogy. We are all keenly aware of the profound impact pandemics can have across human populations. And today, Andy will be talking about the occurrence and consequences of pandemics in marine mammal species. So with that, I'll turn it over to Andy.
Thank you, Austin. Hi, everybody. I'm sorry that we're not all together in person, and this is an odd experience to be giving a seminar by Zoom. Let me start, though, by hoping that everybody's well and hunkered down and wishing everybody a happy Earth Day. It's the 50th anniversary of Earth Day. It's a kind of a big deal. And I'd also just like to recognize the contribution of my wife and muse, Kim Urian, who has helped me think through this topic and helped me considerably with the presentation. So a couple of weeks ago, we decided that we would put all the Marine Lab seminars online. We canceled all of our visiting seminars and had folks from the Marine Lab step up and volunteer to give seminars over Zoom. So I volunteered and I thought it'd be kind of fun to give a seminar about marine mammal die-offs. I didn't think much more about it until a couple of weeks went by and I started to think about the talk I was going to have to give. And I came to the recognition that I really don't know very much about marine mammal die-offs, which is a bit of a problem when you have to give a seminar. Anyway, I put that unsavory thought aside until earlier last week when I actually started to put the slides together, and this kind of mounting sense of fear and anxiety started to rise in me because I really started to recognize I didn't know very much about marine mammal disease and die-offs. And again, I kept working on the talk, kind of put it aside, until last night when I started to get emails from friends and colleagues who I love and admire, who really do know something about marine mammal die-offs and who are on the Zoom call right now, which is why that cartoon is up there. So this is kind of your welcome to science hell, because I'm going to tell you things that you know way more about than I do. And I apologize for that in advance. Secondly, there are some funny images here because we're all living through a horrible experience. So Kim's responsible, as those of you who know her won't be surprised, for all the amusing imagery. And third, which is directly related to number one, this is going to be a pretty short seminar because I really don't know very much about marine mammal disease. So, the other positive part of that is that it's going to be a non-technical seminar. And I wish I could say that I had designed that specifically, but it's really because I don't know very much about marine mammal disease. So it's going to be very non-technical. I'm not going to show any slides of gross dead animals or histopathology slides that you can't interpret. It's going to really be from a different perspective. So let me tell you about that perspective. This picture is of me practicing extreme social distancing. This is 35 miles east of Argon Inlet on the RV Barber with my incredibly awesome field crew. And you can see I'm even practicing social distancing on the boat. I'm 6 feet away from Heather, who's working busily away while I'm not doing anything, as is usually the case. But the point of the image here is that when I think about marine mammal die-offs, I think about them as a conservation scientist and really a conservation biologist. So I think about numbers. I think about animals dying and animals being born. I think about the effects of disease and epidemics on conservation trajectories. And I think about interventions. What can we do to improve the likelihood of population recovery with conservation and species at risk? And what does disease mean for that? How do we have to manage that? So that's the lens that I'm looking through this issue at, and that's what I will be talking about today. So let's start in 1931, which is always a good place to start. And those of you who've taken an ecology course will have run across this gentleman, Charles Elton, one of the founders of the field of modern ecology, who quite literally wrote the book on animal ecology. And Elton was very forward-thinking, brilliant man, spent most of his career at Oxford. And I'm just going to read the first couple of sentences of this seminal paper on the study of epidemic diseases amongst wild animals, because it really kind of sets the stage. So Elton in 1931 wrote, "Fluctuations in numbers of wild animals are of importance, both in pure and applied ecology. The present paper is a contribution towards coordination of the scattered work that has been published on one phase of the main problem. Although a considerable amount of work has been done on epidemic diseases among wild animals, it has mostly consisted of sporadic and independent pieces of research. And one of the objects of this paper is to show the value and importance, and in fact the necessity of widely coordinated studies in obtaining further advances in the subject." And I'm happy to say that when we think about epidemic diseases or epizootic diseases in marine mammals, there really is a very well-developed field and a whole bunch of experts some of whom are actually on the Zoom call right now. So we're fortunate that we've learned a lot since 1931, and we know that disease can be a really important influence in the regulation of animal populations, including marine mammals. So first of all, a little terminology. Just as in animal postmortems, we don't refer to them as autopsies, we refer to them as necropsies. So the same goes for epidemics. We don't refer to them as epidemics in animal populations, we refer to them as epizootics. And pandemics we refer to as panzootics. And I got an email last night from Michael Moore from the Woods Hole Oceanographic Institution, a very good friend and colleague of mine, who chastised me for using the word pandemic in my title. And I told him it was just a hook, and that on slide 5, I had this information, so I wasn't gonna fall into that trap. So Michael, thank you for keeping me on the straight and narrow path. If you're watching, I will continue to stay on that path. Okay, so let's talk a little bit about mammalian conservation. And I just thought I'd give you a couple of examples of terrestrial conservation issues in which disease plays an important role. And these are ones that you may very well have heard of. So first of all, Tasmanian devils, the world's largest carnivorous marsupial, has this weird facial tumor disease, devil facial tumor disease, which is a cancer that's transmissible, and it's transmitted when animals fight. And this is spread, was first detected in 1996. It spread across about 95% of the species range in Tasmania, and it's resulted in local population losses of up to 90%. There are actually two different cancers involved here. There's some good news that looks like animals may be getting resistant to some of that, and there are also very active conservation efforts, including a rescue population that is isolated to which the disease can't be transmitted. But this is obviously a major issue in the conservation of these marsupials. Black-footed ferrets, another highly endangered mammal from the American Midwest, and that species is imperiled by two different diseases, canine distemper virus, CDV, which was present in the population, the wild population when it was discovered in 1981. CDV is 100% fatal to ferrets, but there's a vaccine, which is a good thing. And then sylvatic plague is a more recent disease that affects these animals. Sylvatic plague is a bacterial disease. It's similar, maybe even identical to the bacterium that causes bubonic plague in humans. And the sylvatic plague affects both ferrets and their primary prey, prairie dogs. So there's a vaccine, I think, for black-footed ferrets, but not yet, I don't think, for prairie dogs. Increasingly, though, we are concerned in the conservation community about the human spread of disease or human inducements of disease. And again, here are three more examples from terrestrial mammalian conservation. In 1994 in the Serengeti, the very famous population of lions that Craig Packer studies suffered an outbreak of canine distemper virus, CDV again, which is a type of morbillivirus, and we'll come back to that as we talk about dolphins. That resulted in the loss of about 30% of the lions in the Serengeti. Canine distemper virus was introduced to the lions by domestic dogs. So the encroachment of humans into that population's area, into that population's habitat, led to the transmission of disease from domestic dogs to the lions. And then from those lions, the disease actually spread to other populations of lions and to hyenas and to leopards and other carnivores. White nose syndrome, very famous recent outbreak of disease. This is a fungus that affects many species of bats. I've highlighted little brown bats here because they're one of the species that's most affected. White nose syndrome was first detected in upper state New York in 2007. The fungus affects the activity of the animals, especially when they're in torpor, when they're hibernating. It makes them more active, and that results in energetic imbalances and the morbidity and mortality of many bats. And we've seen this fungus spread throughout North America now, resulting in reductions of 90 to sometimes 100% of bats in some areas. We think that the fungus was introduced by people who had been in caves, in humans who had been exploring caves in Europe, and then brought the fungus to caves in North America, and then the bats, the fungus was introduced to bats that way. Lastly, saiga, which are kind of weird looking goat-like antelopes from Central Asia. You might have seen a few years ago, there was a massive die-off of a couple of 100,000 saiga that died of hemorrhagic septicemia. That was about 2/3 of the population in places like Kazakhstan. These animals died as a result of septicemia caused by a bacterium, Pasteurella multocida, that is a normal part of the bacterial flora of these animals. But the animals were experiencing very unusually warm and humid periods associated, we think, with climate change and global warming that led to that bacteria leading to the septicemia. This last one is kind of the ones that one of the human-induced epizootics that really kind of give me pause and make me concerned for the future. As our climate is changing so rapidly, I think we'll see more outbreaks of disease like this. So here's a picture of a whole bunch of walruses in Alaska. These animals were onshore because there was no ice. Walruses aggregate in large, dense concentrations normally, but in the absence of ice, those concentrations get even greater. So if you think about the potential for a disease outbreak in a population like this, you can imagine that either through physical transmission or respiratory transmission, the potential for disease outbreak is enormous with a species like this. we have seen a large number of marine mammal epizootics. These data are from a paper by Francis Gulland and Ailsa Hall, two other good friends of mine who really should be giving this talk, not me, in EcoHealth. And just to kind of, these are a few I've taken from their paper. And you can see that we've seen epizootics in pinnipeds and cetaceans. We see primarily viruses in this list, all but the leptospirosis, I think here are viruses. The numbers there are all underestimates for the most part, because almost in all cases, they're the number of dead animals recovered, not the total number of animals we think have died. In addition, it's worth noting that some populations are susceptible to repeated infections and outbreaks. So we had a very large outbreak of harbor seals in 1988 in Europe, Phocine distemper, similar to the canine distemper virus, morbillivirus, in which we think 18,000 harbor seals died. And then 14 years later, there was another massive die-off in which about 25,000 animals died. So some populations are susceptible to repeated outbreaks of disease, and I'll come back to this a little later when I finish the talk. So let's talk about something local, something those of us here on the Atlantic coast experienced, and I'm looking at some people who spend a lot of time on beaches responding to the die-offs of dolphins during the epizootic of 2013 to 2015. During that period, something like 1,600 dolphins stranded. And this is a typical example. This is from October 2013, Hammocks Beach State Park, a pretty typical presentation of an animal that died as a result of morbillivirus. The animal's very thin. You can kind of see what we refer to as the peanut head. Animals often have pneumonia, other respiratory issues, and sometimes neurological issues, lesions in the brain. There's a whole series of symptoms. When we look at the rate of strandings, that the outbreak extended from New York State all the way down to Central Florida to Brevard County. When we compared the number of strandings per month or per year in each state over that period of time, there's something like an 8-fold on average increase in typical stranding rates. And you see very large numbers of dolphins stranded in Virginia and North Carolina and in Florida. And this led the National Marine Fisheries Service, part of NOAA, to declare what's referred to as an unusual mortality event, which facilitates the resources and people's time to be devoted to trying to understand the causes of the epizootic. And some of the folks on the call have been intimately involved in UMEs for many years, including this one. So the dolphins died as a result of exposure to a morbillivirus, dolphin morbillivirus. So these are viruses in the family I can't say it. You can read it right there. Morbilliviruses cause measles in humans, canine distemper like CDV, as I said, rinderpest in cattle. So it's a pretty big family of an important family of viruses. As you read down this fact sheet that was put out by NOAA, you can see some of the typical presentation lesions that animals that were affected with. There are also porpoise morbilliviruses, phocine seal morbilliviruses. So this family of viruses is a very important vector for many marine mammals. So how many dolphins died? So it's really, we know that we had something like 1,600 animals stranded, but we know that the number of animals stranding is some small but unknown proportion of the number of animals affected, right? We all want to know about testing with COVID-19, how many people are getting tested, what is the infection rate, how many people have antibodies. So we don't know how many animals died. We know how many animals were presented on the beach. We know that not all stranded animals were recovered. On the ocean beaches, we probably got most of them, but in places like the marshes and Pamlico Sound, a lot of animals could have died that would never have been recovered. And then a bunch of stranded animals were not tested, either because there weren't the resources to do so, or because the carcass was so decomposed that it was not worth testing. The animals had been dead too long. So we really don't know how many animals died in the epizootic. And just as an aside, as you're thinking about COVID-19 and you're thinking about science and you're thinking about the role of science in policy, you should get on Google Scholar and look up Dr. Anthony Fauci. You might not believe in things like the H index, which is a way we kind of measure how many times our work is cited. Dr. Fauci's H index is 220, which is crazy, crazy high. It's 3 times higher than mine. He is a bona fide scientist, and we should all listen to what the hell he says and not listen to other people. That's all I got to say about that. Okay, interestingly, this was not the first rodeo for morbillivirus in the Atlantic. In 1987 and 1988, and some of you are old enough to remember this, there was another die-off. In fact, some folks who I know and love who are on this call kind of cut their teeth on responding to strandings during that period. Initially, when dolphins started dying in 1987, 1988, there were all kinds of crazy rumors that it was dolphin AIDS, or that that medical waste had been spilled into the ocean in New Jersey, and that was what was causing the dolphins to die off. Not true. The dolphins died of morbillivirus in 1987 and 1988, just as they did in 2013 and 2015. And I'll come back to that again a little later on. So folks from NOAA, working with some academic colleagues, did some really cool epidemiological modeling of this outbreak. And they would say, I think, if they were giving the seminar, that we should take all this with a grain of salt, because the data that they had to parameterize the models was not great. But it's still pretty interesting. And again, as we're thinking about COVID-19 and we're thinking about things like R0 and we're thinking about vaccinations and interventions, it's kind of interesting to think about marine mammals and marine mammal die-offs in the same way. So what do we know? We know that the peak of the outbreak occurred off Virginia in August 2013. And at that time, in August 2013, each sick dolphin was infecting 2.7 others. So the R0 was 2.7. That's on average, every dolphin was infecting 2.7. And that means that at those rates, the disease is going to spread very, very rapidly. That waned as the epizootic progressed, and it reduced in magnitude, which is a good thing, because eventually when it gets below 1, that means each sick dolphin is infecting less than 1 other dolphin, and the outbreak will eventually die out. The mean period of infectiousness was about a week, a little bit more than a week, 8 days, and 95% of infectiousness was within about 3 weeks. So not a long period of time. But again, think about that when we think about COVID-19, when we're still trying to figure out what these parameter values are for the coronavirus. So again, so as a conservation scientist, these are the questions I want to ask, right? So we have this epizootic, we've got 1,600 dead animals on the beach. What is the population level effect? And so first of all, we need to know what populations, or in the parlance of marine mammal conservation and management, what stocks were affected. How big was each of those stocks prior to the epizootic? How robust were they? And then how many animals died from each stock? And are any of those stocks that we're particularly concerned about because they were small beforehand, or because the mortality rates were so high that, that they give us pause? Unfortunately, or maybe fortunately for those of us who like to dabble in these kinds of things, the population or stock structure of bottlenose dolphins along the US East Coast is incredibly complicated, and we still don't really understand it. There are a lot of people, both from NOAA and academic institutions and other institutions, who've been working on this for 25 years, and we're still sorting it out. What we do know is that there are stocks of dolphins in coastal waters, in the ocean waters, that move north and south. We call them migratory. Whether or not they're really migratory or not, we could argue about, but they move seasonally. So there's a stock that moves seasonally as far as New York and then comes down to North Carolina. There are other animals that move as far north as the Delmarva and south to Florida. And then 3 other kind of ocean stocks as well, although the structure of those stocks is really poorly understood, and it might be different than it's portrayed here. But we also know that there are a whole bunch of small estuarine resident stocks, 2 of them in North Carolina, one in northern North Carolina that extends kind of up into almost into the Chesapeake, maybe right into the Chesapeake a little bit, and down to about the New River. And then a southern North Carolina estuarine stock, which is quite small, that extends from about Beaufort down into South Carolina a little bit. So what are the sizes of those stocks? Thanks to work from NOAA and from academic colleagues, we had a pretty good understanding of how big those stocks were prior to the epizootic. So the 2 migratory stocks, which I'm going to focus on, the northern migratory stock which was surveyed for the last time prior to the epizootic in 2011. Pretty robust, 15,000 animals. CV is the coefficient of variation. It's just a measure of variance scaled to the mean. So the higher that is, the more uncertain we are. We like to have a CV of like 0.2 or less. Southern migratory stock is also pretty big. So taken together, there are about 20,000 animals in those two migratory stocks. So that's good. Those are robust stocks. The two estuarine resident stocks are much smaller. The northern North Carolina estuarine stock, 800 or so, and the southern North Carolina estuarine stock, very small, 188 animals. The migratory stocks are surveyed using planes, doing line transect surveys. The estuarine stocks are surveyed using photographic capture-recapture techniques, in which we actually use the photographs of individuals to capture them and then recapture them in subsequent surveys. So we've got two big migratory stocks and two small estuarine stocks and one very small estuarine stock in southern North Carolina. And I'm not going to talk about them just for the sake of brevity today, but there are a whole bunch of other small estuarine stocks as we move from North Carolina south into Georgia, South Carolina, Georgia, and northern Florida. And again, unfortunately, we wish we were living in kind of dolphin CSI world, right? Where you could wave something over a dolphin or take a little skin sample and go, ah, this is a southern North Carolina estuarine stock, because it's a perfect genetic marker that only those animals have. The world, unfortunately, is not like that. So there are very good molecular biologists like Patty Rozelle from the Southeast Fisheries Science Center who have been spending quite literally decades trying to find molecular tools so that we can assign individual animals to particular stocks. That is not yet possible. We can use assignation tests to look at haplotype frequencies to look at the likelihood that a particular dolphin belongs to a particular stock. But we can't say for certain that this dolphin that my friends and colleagues from UNCW responded to belongs to a particular stock. So that really kind of complicates our ability to assess which stocks were affected and how much they were affected. This is a plot that shows you the cumulative distribution of stranding starting in July 2013 in the panel to the upper left. And each yellow and black dot is a stranded dolphin that tested positive for morbillivirus. And in each panel, it's cumulative. So the dots on the top left panel are still there in the middle panel and all the way down to the bottom right panel. But you can see that over time, the epizootic, which started in the northern part of the range of the species, extends into the mid-Atlantic, into the South Atlantic, and eventually down to northern Florida. And Ani, if you're watching, you can see that there's a dot up there in the Chesapeake Bay. So there was a morbillivirus case all the way up in your study area. So this suggests maybe that the migratory animals were more likely to have been affected. And Patty Rozelle's work on molecular assignation tests seems to confirm that the animals that were tested were more likely to be from the migratory populations, which is a good thing because they're the big ones, right? This is from the epidemiological modeling paper. It's a different way to look at the spatial and temporal patterns of those strandings. So we're looking at latitude here. So Virginia is like 36, 37, something like that. We're about 35. And the day of the outbreak. And you can see that this shows you that kind of very nicely the temporal and spatial patterns of the disease progression. So it starts up there in Virginia with that really intense outbreak, and then it spreads down to Florida, stays kind of south in Florida, and then the next spring it moves back up into the northern part of the range of these animals again. This all makes sense because what we know is about the migratory dolphins is that this is a picture from one of Kim's pictures, I think, from the Outer Banks here. These animals are typically found in very large groups. So when we see estuarine animals, we often see small groups in 5, 10, 15, 20 maybe. The migratory dolphins can often be in 20, 50, or hundreds. And so they're, again, in very dense groups, to very dense aggregations, which facilitates transmission either through physical contact or through respiratory transmission because the animals are all breathing together in very close quarters. So NOAA has been working to try and understand what the impact of the unusual mortality event, the epizootic, might have been. And these are, this is taken from Lance Garrison, one of his reports. He's a stock assessment biologist in Miami from the Southeast Fisheries Science Center. And these are surveys of pooled coastal bottlenose dolphin abundance. So this is from pretty much from New Jersey all the way to Florida. It's not broken up into individual stocks. We don't really have a good, we don't have a good handle of the effect on those individual coastal stocks quite yet. But you can see that these are the estimated numbers plus confidence limits around those point estimates, 2002, 2004, 2010, 2011. So if you look between 2011 and 2016, you can see a very dramatic reduction in the number of dolphins. So from this, it looks like the mortality event from 2013, 2015 resulted in more than half of the dolphins in the coastal area, and that includes the northern and southern migratory animals. More than half of those dolphins died, which is a pretty amazing thing when you think about it. It's not something that I think we talk about enough. We just had a disease outbreak that killed half of the dolphins in the coastal Atlantic waters. And you don't hear about that very often. And I think it deserves more attention than we give it to. This is a slide that Kim provided to me. So we can't use molecular techniques to assign individual dolphins to a particular stock, but we can use other markers. So Kim curates the Mid-Atlantic Bottlenose Dolphin Catalog, which is a catalog that includes images of individual dolphins, like the one here, from New York all the way to Florida. Almost every organization that works on bottlenose dolphins along the Atlantic coast contributes images to the catalog so that we can see where our dolphins go. And it's a great collaborative venture. Kim's the curator, but it's the effort of literally hundreds of people over decades of time. So one of the things that Kim did, she used kind of really cool detective work to try and figure out where some of these well-marked individual dolphins had been before they got sick and died. So here's VGT, that's the VGT 308. VGT are the initials of Vicky Thayer, who did her PhD here at the Marine Lab, who now coordinates the the Marine Mammal Stranding Network in the central part of the state. This animal died in October 2013, a mature male, 283. You can see a picture of there in the middle, pretty typical, really skinny, typical morbillivirus animal. The animal had a really nice distinctive fin, and Vicky took a beautiful picture of the animal after it had stranded. Kim was able to match that picture to 4 catalogs. So pictures of the animal when it was still alive between 2002 and 2013. And the locations of those images are shown here on the map on the left. And they're color coordinated. The Maritime Museum, so Keith Rittmaster and Vicky Thayer, who have been studying dolphins here since the late Pleistocene. I think they kind of started everybody on studying dolphins a long time ago. Our catalog, the Beaufort NIPS Lab catalog, and then the catalog from the OBX Center for Dolphin Research, which is run by Jess Taylor, who's a CM grad from our program who works up in the Roanoke Sound. So you can see where this mature male, ranged widely within Pamlico Sound. This is a classical northern North Carolina estuarine stock signature. So this tells us that at least some estuarine dolphins were affected. We don't know how many. There are several cases like this, but obviously you can't match every dolphin, right? Some animals are too decomposed. A lot of animals died when they were young and their fins were not identifiable, so we can't use this technique to assign them to a particular stock. But we know that some of these small estuarine stocks were affected. So let's think about context a little bit and think about what this means for those populations. So these are 2 models of basic mammalian population growth, actually population growth of any animal. Exponential growth, change in population over time, is equal to the rate of population growth times the population at any time. And those of you who've taken my classes have seen this probably until you're sick of seeing this. But we know the populations, nobody but economists believe that populations can grow like that indefinitely, right? So something eventually is going to limit population growth. So we assume that mammal populations grow according to this logistical growth model, where their growth is initially very rapid, and then some factor, space, water, food, will limit their population growth until they achieve the carrying capacity of that environment. When we think about marine mammals, if you think about your old ecology classes, think about the old concept of R and K selection. We think about marine mammals as being classically K-selected, that they've evolved to survive and thrive near their carrying capacity. And so they're selected for factors that let individuals thrive in a pretty intense interspecific competitive world. But what epizootics do is to drive those populations down away from carrying capacity, down that curve. And that changes kind of the selective factors that might be advantageous, especially if populations were exposed to epizootics on a repeated basis. So just think about this. Think about the fact that we are driving those populations from somewhere near carrying capacity down to much lower levels. We just saw that maybe populations of dolphins in the Atlantic were reduced by half, for example. So that's taken them down, if they were at carrying capacity to begin with, down to about half of carrying capacity. And then that brings us to how we manage and how we intervene and what we do. So the Marine Mammal Protection Act of 1972, which regulates how we conserve and manage bottlenose dolphins in the Atlantic and all other marine mammals in the United States, has two fundamental goals. One, to maintain stocks of marine mammals as functioning elements of their ecosystem, which is a very noble goal that we don't actually manage on, but it's in the statute, in the wording of the statute. And secondly, the way we actually do manage in the real world is to maintain stocks of marine mammals at what Congress wrote were their optimum sustainable population levels, or OSP levels. So what the hell is OSP? That's a good question. That the two agencies that have to implement the MMPA, the Fish and Wildlife Service and NOAA, had to figure out. So they defined, the two agencies back in the 70s defined OSP as the range of population, and this is from the Federal Register, the range of population sizes from the largest supportable within the ecosystem, that's the carrying capacity, K, that we saw on the previous graph, to that which results in maximum net productivity. We think that if that logistic growth curve is nice and symmetrical, populations of marine mammals should have maximum productivity. That's the maximum number of individuals added to the population each year at about half of carrying capacity. So in practice, what the MMPA tells us, the way we interpret the language of the statute, we should try and keep marine mammal populations at no less than half of carrying capacity. Anywhere between half of carrying capacity and carrying capacity is good. And if we get below half of carrying capacity, that's bad. And we refer to those populations as depleted, and we spend particular attention to try and manage and conserve them. So we can imagine these epizootics might be pushing populations from a good conservation status within their OSP level to a poor conservation status below their OSP level. And this has complications for the way we manage in practice. Kim looked at this slide last night and said, oh my God, you're really going to show that? It's not really as bad as it looks. So it's the same graph. It's the OSP graph with maximum net productivity there in the level. And then there are two lines. Those lines are PBR. I'll explain what PBR is in a second. So when we think about removals, we think about managing removals. So think about managing the number of animals that are killed in a particular marine mammal stock as a result of fisheries interactions. The Marine Mammal Protection Act gives us explicit guidance. There's a biological reference point, the potential biological removal level, which is the maximum number of animals, importantly, not including natural mortalities, that may be removed from a stock while allowing that stock to reach or maintain its optimum sustainable population. And the PBR concept and the equation was developed as a result of very intensive modeling I won't go into now. But if you, the take-home message is if you limit mortality, anthropogenic mortality, human-caused mortality to below PBR, any marine mammal stock should equilibrate at its OSP level, above half of carrying capacity. This is the PBR equation. It's not really very important other than to say that PBR scales directly with population size. And that's why those two lines that are intersecting that yield curve there, if you think about this as kind of an economics yield curve, PBR is a direct function of population size. As population goes up, the number of animals you're allowed to remove goes up. As population size goes down, PBR goes down. So that means if you have an epizootic that kills half of the dolphins in a stock, all of a sudden you're only allowed to take at most half of the number you could have taken as a result of fisheries interactions or some other anthropogenic cause. So it means we have to manage much more conservatively. And that's what happens, right? So the population goes down, PBR goes down as well. Along the Atlantic coast, and something that I and many other people on this call have been working on for a couple of decades now, the major threat to bottlenose dolphins is fisheries bycatch. And there are a variety of fisheries that bottlenose dolphins interact with. Gillnet fisheries like this one from our friends in UNCW are the primary cause of fisheries bycatch. So that means as we're trying to manage and trying to work with our colleagues in the fishing industry to reduce the number of animals that are killed each year, when we have these epizootics, that just makes everything so much harder, because the number of animals that we can legally authorize to be removed from each stock is being reduced by these epizootics. And interestingly, some animals that were killed in fisheries tested positive for morbillivirus. This is another one of Vicky's animal. This is an animal that died in a stop net on Bogue Banks in the fall of 2013. So the animal still got some of the stop net gear on it. If you know, those of you who've been here in Beaufort in the fall will have seen stop net fishery. It's directed at roe mullet in the fall when the mullet come out of the estuaries and migrate along the coast. So this animal died as a result of a fisheries entanglement, but it tested positive for morbillivirus. We're raising all kinds of questions like, are sick animals more likely to get entangled? How do you score this? Is this an animal that died as a result of the epizootic or an animal that died as a result of bycatch? How do we deal with issues like this? Okay, I'm about done. So just a couple of final thoughts just to maybe get you thinking a little bit. And these are absolutely not fully formed because my thinking about these questions is not fully formed at all. It's kind of half-baked, but that's okay. It's better than not baked at all. So for bottlenose dolphins, we had an epizootic in 87 and 88. We had one in 2013 and 2015. And so one of the questions that immediately arises is, will outbreaks occur or reoccur each time when population level immunity wanes? If we look at the age structure of animals that died in 2013, 2015, most of the animals that died were not alive when the 87, 88 epizootic hit, which meant that they probably didn't have any immunity to that. So does that mean every 20 or 25 years, we're going to have another outbreak? And that means that the population gets driven down each time that occurs? And if that's the case, where is the reservoir of morbillivirus? It's not in the population, otherwise we'd have herd immunity to it. So some folks, after the 87 and 88 die-off, suggested that pelagic cetaceans, perhaps longfin or shortfin pilot whales, for who we know carry morbillivirus and have outbreaks of morbillivirus, maybe those animals are where the reservoir of dolphin morbillivirus is. And occasionally, these coastal migratory dolphins will come in contact, contract the disease, and then you get another outbreak. So if that's the case, that we're going to be seeing this kind of on every couple of decades or so, we're going to have another outbreak. More generally, though, as we think about disease and marine mammals, are some populations regulated by repeated epizootics? Some populations never really live very much around carrying capacity. That would be a really strange and new way of thinking about population regulation in marine mammals. But again, if you go back thinking about that picture of the walruses, the walrus, maybe that's not such a crazy thing, right? If you think about harbor seals or elephant seals or gray seals aggregating in really dense aggregations on breeding rookeries or haul-out sites, maybe that's something we should expect these populations to experience. And then on a related factor, has that always been the case? And it would be really interesting to know more about the history. We really don't know much about historical outbreaks before the 60s or so. Like some of the factors I showed you with terrestrial conservation issues and disease, is this mediated by some human activity? Are we doing something? Or is climate change or something else affecting the likelihood of these epizootics? And then lastly, how do we manage removals from populations that are experiencing repeated outbreaks like this? And so there's kind of a lot to think about. And as we think about COVID-19 and we think about where we are, hunkered down in our homes, hoping for a new normal, thinking about how things are going to get better, what do we think about? We think about vaccinations, right? Well, marine mammal scientists beat you to the punch. So there's a person on the right, Michelle Barbieri, who's the veterinarian, the lead for the Hawaiian monk seal program now, a graduate of the UNCW program, one of many of us, one of our favorite people. She's vaccinating a monk seal in Hawaii against morbillivirus. So we have about 1,400 or so monk seals in Hawaii. They're a critically endangered population. The population has been declining, although the decline has started to stabilize. The monk seal conservation program, those of you who've taken my classes know that I think it's one of the most forward-thinking conservation programs anywhere in the world, led by really smart, visionary people like Charles Lettman and Michelle. And so they've actually looked at which monk seals interact more frequently with other monk seals, targeting individual monk seals to vaccinate to prevent the outbreak of a disease like we saw in Atlantic bottlenose dolphins. So maybe at some point in the future, some of you guys who are students now who are watching this will be out there vaccinating bottlenose dolphins against morbillivirus, or vaccinating other populations or mammals in the same way. And hopefully by that time, we'll also have a COVID-19 vaccination. Okay, I want to end again by apologizing to all my colleagues who actually do this for a living and who should really be giving this talk, but also to thank them, to all of the folks who I have worked with for decades, who have spent their lives trying to understand the demography, ecology, and health of these animals, and particularly to the folks who are on the beaches, our first responders, without whom we really wouldn't understand any of this at all. And thank you to all of you for listening. And I would be happy to try and take any questions now if Austin can facilitate that.
It's Lisa. I'll let Austin facilitate if you like.
Okay, you can do it. I don't care who does it.
No, I don't mind. I was just going to say there's a bunch of people on the YouTube live stream. People were having a hard time getting in to the meeting originally. So even though there's 73 people in the Zoom link, there's over 100 on the YouTube stream. So there you go, Andy, you're popular. For those of you who are familiar with Zoom, we've been trying to use the hand raise function. The other way is that if everyone has their video off, then when they turn their video on, we know that they want to ask a question. So I'll open it up for questions. D. Allen has their video. No. Samantha has her video on. No. D. Allen. Okay, everyone take a minute and put their video on or off, depending how they want it.
Hi, it's Dee. I was not going to ask, but then I was like, actually, I do have a question. And this might be, maybe someone in the audience can answer this. I was wondering about, is there any immunity transmission from a mom to a fetus that we know of?
I don't know the answer to that, Dee, but I bet somebody else does.
Thanks, Andy.
If you do, speak up. Maybe we don't. It's a good question. I suspect I'm going to be saying I don't know a lot, just so you can get ready for that. Other questions?
Hey, Andy, this is Tara.
Hey, Tara.
So you had my juices flowing thinking about if this has happened before 1987, 88, is this a regular occurrence? Do you know, has anybody looked through news articles? I was thinking maybe like Virginia Beach might be a good place to start, looking to see if they reported, back in the 1800s, if there were unusual spikes in strandings.
Yeah, I don't know that. I would think if anybody did, probably WAM or DAP would know that. So I don't know if they want to weigh in. I've never seen any indications of that. We know that there was a very large directed harvest of bottlenose dolphins around the turn of the century and prior to that. But WAM and DAP, do you know if there's any evidence for that?
Great question. Actually, Jim Mead he hunted around, of course, since he was back there then. There is I think there's one story in the Washington Post of a unknown number of dolphins hitting the beaches in Virginia in the 60s. So of course the timing is exactly right, but no other information than that. So that's the best we know right now, but at least there is evidence of another 25-year mortality or potential mortality event in the 60s in Virginia.
Yeah, I wish we had better historical records. And I wondered about that when I was putting the seminar together. Like, would we know in the 30s or 40s if there were a bunch of dolphins dying on the beach? I'm not sure that we would. I'm not sure that anybody in Hatteras, or even if they noticed in Hatteras, if that news would have got anywhere, that it would have been to a newspaper in DC or in Raleigh.
The only other thing I'll add here is there's a pretty good paper. Terry Rolls did a paper looking at the immunity of dolphins, and she led it up to, in the 50% number, she basically predicted that they were seeing 50% of the animals showed no evidence of immunity to this, and she said it was only a matter of time then. So that was predicted in the
2011. Or
2012
yeah.
paper.
Right.
So it's starting to follow through. The 50% mortality and 50% infection is following through like most other large mammal populations.
Right. Another question.
Andy, this is DAP. That was such a wonderful presentation. And I'm just looking at Michael McGowan's chat question, which was something that I was going to think about. And maybe dolphins, bottlenose dolphins would be kind of difficult, but museum specimens and the ability to potentially detect viral specimens in older museum specimens might be an interesting way of going through it. And I think that's a great idea. I don't know how CSI that sort of technique is, but that might be a good way to think about it.
Yeah, that's a really good idea. Yeah, way outside my area of knowledge, but I wonder, yeah, that's a really interesting idea. Again, I don't know.
I saw Parker and then Jillian.
My question is, so I'm thinking about the vaccinations in monk seals, and that seems a little bit more accessible since they are land-based part of the time. And I know that there are health assessments in the states further south, but do you foresee, what challenges do you foresee with creating a vaccine for marine mammals, or has your research given you any indication of that yet?
Hi, Parker. It's nice to see you. Yeah, no, certainly not something I would do, but as you can imagine, with a really small population, right? So we know how to catch bottlenose dolphins. We know how to do health assessments. We've done them here in Beaufort. They're done routinely in Sarasota. So you can imagine if you had a population that was of particular concern and you had a vaccine that worked, maybe you could trial a vaccine with captive animals or animals in human care, that you would be able to do that at least for estuarine animals. So I think if you ever got to the point at which something was of that level of conservation concern, like it is with monk seals, that you would be able to capture and administer a vaccine that way. So think about other small populations of marine mammals that are readily captured. I think that's doable. But you're right. I mean, monk seals are pretty easy. Those of you who've seen monk seals, the one thing they're really good at doing is sleeping on beaches. And so you can kind of creep up and poke them with the vaccination stick. So, but yeah, clearly harder with cetaceans, but not impossible.
So you would foresee this being more of a solution for coastal communities as opposed to pelagic species?
Yeah, pelagic would be hard because you'd have to do it remotely. And again, there are folks who tried to administer things like sedatives. So Michael Moore and other folks on this call. It's really hard because you have to get up close to an animal and get through the blubber, and you have to estimate body mass. And so all those kinds of things are really tough. So unless you can capture animals, it would be hard. But again, think about 20 or 30 years from now, who knows? You guys are all smart. You got your careers ahead of you to figure out how to do things like that.
Thanks.
Yeah.
All right, other questions?
I'll ask mine.
Hi, Jillian.
Hey, great talk, Andy. So obviously I'm sitting here thinking about stress. I think maybe we all are actually. So yeah, I mean, we know that stress and contaminants can affect the immune response. So I'm wondering if there are any concrete examples of animals that are more exposed to anthropogenic disturbance having higher mortality rates in response to some of these diseases that you've talked about.
Yeah, that's a really good question. So increased stress, less resistance, more susceptibility. Man, I don't know. Maybe somebody else on the call knows, but the other thing I wonder there too is, for example, we suspect that animals in some places along the US East Coast, but we know that they have very high burdens of persistent organic chlorines, persistent organic halogens. So in those animals, like the animals that Brian Balmer studied along the coast of Georgia, those animals more susceptible because they have such high burdens. So that's a different type of stressor, but those kinds of stressors, I would think would influence the immune response. And again, I don't know, maybe if somebody else wants to weigh in on that they should, but I'm just waving my arms around, not really knowing anything about that. But we do know that some of these populations have very extraordinarily high levels of burdens. And so you'd be particularly concerned about them. It's a good question.
Thanks, Sandy.
Yep.
Ann and Bill, did you want to go?
Yeah, thanks. I can't speak to that, but I do want to throw out the question of PBR and management, and how robust is PBR and management strategies, current strategies to deal with a 50% reduction in a population? Is it something that's functional?
Yeah, well, it's not very functional right now because we still can't assign individual animals to stocks. So when we have a dead dolphin on the beach, it's been killed in a gill net off Cape Hatteras, for example. We still don't know which stock it belongs to unless we can identify it from its dorsal fin, and Kim can do her magic. So that's a fundamental problem. I really think I think that if we had populations that were repeatedly exposed and repeatedly driven down as a result of epizootics, we would want to think hard about how we're managing them. And I like I said on that slide, those questions are not fully formed. And I really think we kind of need to think about that a little bit. I don't have an answer to that, but it changes the status of the stock, for example, from within OSP to depleted. And that would mean that it's much more likely that any takes from that stock are going to be strategic, that they'll be above PBR because the PBR threshold will have come down. But is it really fair to penalize fishermen who are doing the same things, catching a few dolphins when the population of dolphins that are being affected all of a sudden is dramatically reduced in size? So I don't think anybody's thought through that question very carefully. At the same time, the intent of the MMPA is to protect marine mammals and to maintain marine mammals as functioning elements of their ecosystem and within their OSP. So it just means that we will have to work harder, I guess, to manage those mortalities and try and minimize them, which we should be doing anyway. But, yeah, makes it harder. I'm not seeing the chat, so I can't see if there are questions.
We have some comments in the chat, but I don't see any specific questions.
Hopefully they're not. Hopefully they're not vituperative comments. Well, I think if not, then this is a good time to wind it up. I just want to say thank you, everybody, for coming out today. And please, everybody, be well, be safe, hunker down. We'll all get through this on the other side and get to spend time together again in person. And I'm looking forward to that. So thank you, everybody.
Thank you, Andy.
Bye. Thank you.
Thanks, guys.
Thanks, Andy.
Thank you.
Thanks, Andy.
Hey, Lee, bye.
Thank you.